the nitty-gritty of things, I would like to give you just a little bit about my educational background, just to let you know what my journey has been. And again, to emphasize on what Bumisola said, that I don't really have a formal training for data science or even IT, but everything I've done is all self-taught. So things you gather on the internet, you teach yourself, you grow from it. So I completed my PhD not too long ago in cell and molecular biology, mainly plant biology, and I'm currently working as a postdoctoral fellow where I use computational and molecular biology tools to try to solve the food security problem. I'm going to get to that problem much later, where I will use it as a case study so that you get a better appreciation of what things are. So before we go any further, it would be very nice for us to get some key terms down, just because we use things interchangeably in this field. When we talk about data science, we're talking about bioinformatics, which some of you might have heard of, some of you might not. Same thing, computational genomics, same thing, genomic data science. And this definition that I have on my slide does it justice, where bioinformatics and all of these data science things are like a marriage between biology, computer science, and information technology. So all of these disciplines come together to form one single discipline, which we then call bioinformatics, or some people would rather call it computational genomics, computational biology, genomic data science. This word map over here does things, I think it does things justice in the sense that most of you would see languages you're familiar with. So computational languages, coding languages, Python, R, which I'm a huge fan of R. I know some people have their peeves about it, but I love it. It makes very, very nice, crisp images. You're able to do biostatistics on it. In fact, you hardly see any computational biologist that doesn't use R. Neither do you see any biological science article that deals with a lot of data that doesn't incorporate R into it. So R is very useful. It's an open source tool. I'm going to talk a little bit on it towards the end of the presentation, but this is something I would also like to throw out there right now. So we know that when we are doing anything science related, you got to have a hypothesis. So all of these come together. All I'm trying to say is all of this come together to form a single discipline. Before I also go far, I would like to introduce a few things. Talking about bioinformatics, we're talking about data science. Where do all these data come from? Well, they come from analysis and interpretation of biological data, which is just a fancy way of saying they are nucleotide and amino acid sequences. For those of you who aren't biologists, you would know that, or you should know that nucleotides are the building blocks of your DNA, while amino acid sequences are the building block of your protein. So all these things are sequenced. Again, these are things that we could spend hours talking about, but just for the sake of this talk, just know that all of these things are sequenced, and then the data we get from them is what we apply computational work to be able to make sense of things. In addition, what most of we biological data scientists do is to develop new algorithms and statistics so that we can find relationships among members of large data. So you have a huge data, you want to make sense of everything. I'm going to be walking through a practical example later on in this talk, and that would give you a sense of how you can draw relationships between your huge and gigantic data sets and make sense of it all, draw the attention of your audience to the things you're very interested in sharing with them. And I would also like to point out that this analysis network, looks very impressive. It was made through R, just so you know that you can do cool things with R, and also let you know why I'm a sucker for R. That being said, another thing I would like to introduce you to before we get into the case study I'll be presenting today is areas where bioinformatics can be applied. So most of you could be thinking, okay, she's talking about biology, she's talking about data science, how can we apply it? You can apply it to rather a few things. You can apply it to genomics, which could be used for genomic prediction, sequencing data analysis. That's one of the things I'm going to touch on today. You can also apply it to proteomics. This could be of interest to those in the biochemistry field, where you want to study protein interaction, protein modeling, interaction of drugs that have been designed by pharmaceutical companies and all of that. All of these things are useful, and they can be used in a bioinformatics point of view as well. System biology is another application where you're looking at things from a whole system. So from an whole organism point of view, you could look at gene enrichment, pathway analysis, where you want to know what things are interacting, and not only how are they interacting, what's the pathway, what's the sequence of these interactions. You could also apply data science to phenotypic data, where you take a data or a picture, very amazing picture of something with your camera, but some things aren't quite right. You can write an algorithm to tweak things around, make your images look better, either for publication purposes, for presentation purposes, or whatnot. So all of these just to say that bioinformatics, data science, whatever you want to call it, is very useful, is very applicable in the area of pure science research as well. So now I would introduce my case study using and mainly talking about sequencing, data analysis, and gene enrichment. So out of all of these, we're only going to touch on 2 today, again for time, and just because I think this would allow you guys to have a better appreciation of things. So the food security problem, why is this important? Well, we know that the human population is on the rise, and it's been projected that in order to sustain this population by the year 2050, we need to produce approximately 75 to 100% more food. Again, 90% of our daily caloric intake comes from plants. Some of us eat animals, some of us, are vegetarian, but majority of our food comes from plants and crop plants. And the main problem is, while we need to grow more plants, we're losing land to grow these plants. We all can attest to seeing large area of field where once filled with grass and trees being cut, being cleared for houses and industrial companies or industrial structures to be erected. This is a huge problem. In addition, there's a change in environmental condition where water availability is becoming an issue. You get too much water in some regions, you get flooding, crops aren't doing so well. You get little water in some regions, you have drought, again, crops aren't doing so well. So scientists and agriculturalists over the years keep battling with how do we increase yield? How do we make these crops grow better? And one of the things they utilize is pesticides. Because not only do plants face all the major challenges I've mentioned before, crops are also being attacked by pests. Most of you have probably heard in the news of the recent locust attack in Kenya, I would say, where field of crops are just rendered useless in an instant. So farmers continue to use pesticides, they continue to use all these things. Another way through which they increase yield is by fertilizer, the addition of fertilizer, chemical fertilizers, and this is a problem. Not only are crops getting less efficient in using these fertilizers, and these fertilizers remain in the soil. When rain falls, they get run off into other water bodies, they kill aquatic organisms. It just becomes a nasty problem. So what we are trying to do in our area of research is to see if we can come up with microorganisms that are beneficial to the plants. So these microorganisms could be bacteria, could be fungi, but for the sake of this talk, I would only focus on the bacteria side of things. And all of these are called biological additives. These biological additives, bacteria, they help promote plant growth, and they also take away the old nasty problems caused by fertilizers. Another thing that farmers do is by practicing crop rotation. If you guys can raise your hand or just put something up on Zoom if it's something you've heard of, if you've heard about the practice of crop rotation, so that I know, yeah, okay. Well, crop rotation is a practice that has been on for centuries. Farmers, both rural and industrial farmers, they practice it, where the whole idea is to rotate your crops every year. So assuming a farmer plants corn in this year, next year, that farmer wouldn't want to plant corn on the same plot, just because not only have the corn plants, utilized the nutrients that corn crops do utilize, the field is also prone to have pests that are particular to corn. So the following year, the farmer could plant soybean, could plant a leguminous plant, a soybean or groundnuts, just to switch things up, because these other groups of plants will utilize different kinds of nutrients from the soil and would also disrupt a particular pathogenic cycle. So either pest or, well, yeah, pest or pathogen, whatever it is, it would stop that cycle and then things can be done. Just let me collapse this right here. So one of the questions we're interested in solving and in also answering is, what are the effects of crop rotation on indigenous microbial communities? This is a practice that has been on for centuries, not even decades, centuries, but nobody has seen how this sole practice affects what is happening below ground. Is it affecting the good microbes? Is it affecting the bad microbes? What is it doing? And then we want to know if we can isolate some of these microorganisms and use them to improve crop performance. This is where we're going to delve just a little bit into how data science was used to answer some of these questions. So the first thing we did was to come up with an experimental design where we selected a field, a farm plot that had different rotational systems, 3 rotational systems. One year corn was grown, the following year soybean was grown. In another plot, canola was grown the previous year, soybean was grown the subsequent year. And canola, if some of you aren't familiar with it, it's a cash crop here. It's a crop of economic importance. It's also the crop where canola oil, maybe you've heard of it, is extracted from. So it's a huge crop. Then our third experimental system was a monoculture regime, where soybean was planted in one year, and then the subsequent year, soybean was grown. All of these had their important replicates. We all know as scientists, or those of you who do experiments know that replicates are very essential to be able to draw or make sense, draw conclusions from your data, make sense of the data. So all of that was done. I wouldn't bore you too much about the details. And then we came up with a pipeline of how we're going to do things. Collected our samples, we performed DNA extraction, well laid out protocols of all of this online. It's been established. If you're really curious about how all of this is done, just do a little digging online. You would find tons of information on that. Next, we prepared our libraries and we did quality control and pooling. So when you talk about library preparation, these are just extra steps in your experimental system where you just need to make sure that things are right. So your DNA sample, you add the essential primers and all the things you need to add before you do your Illumina sequencing. Illumina sequencing is a powerful tool for some of you who aren't too familiar with all of these. It's a powerful tool because it allows you to do a lot more with your samples. You can do things on a ground scale of things. And what we really want to do from all of this, what we really want to get from all of this is to know the sequences of things. So to know the genetic information contained in all of these samples. When we did the Illumina sequencing, this is what we got. Tons of information. We're talking about reads as much as 20 million reads. It's gigantic. There's no way you can power through this data manually. Like I said, tons of data. So yeah, data science to the rescue. That's how we're going to solve the problem of these amounts of data. So based on this talk, I would only focus on one of the spreadsheets I've been working with. It's a spreadsheet that looks like this, where you have data as much as 350 pages on your Excel spreadsheet. How do you make sense of this? It's ridiculous. So decided to just focus on a subset of things where we're looking at 812 million reads. To some of you, this could still be mind-blowing. It's mind-blowing, it's quite impressive, but it's nothing compared to the total of things we got. We also have number of bases as much as 160 billion. This is where KIME comes to the rescue. So KIME is an acronym for Quantitative Insight into Microbial Ecology 2. And with KIME, what we want to do is we want to identify differences in our samples. So how are our plots different from each other? What is different in the soybean plot that is not in the corn plot, that is not in the canola plot, bacteria-wise? Again, like I told you, for this study, we're only focusing on bacteria. And once we're able to identify what is different, can we identify who is different. So which species are there? Sorry, my slide just keeps doing things. So sorry if I need to go back. So once we identify all these differences in community composition, we want to know how can we apply it? What's the application? What's the functional analysis? What can be done? And I'll try to walk you through all of these processes today before the end of this presentation. So how does crop rotation affect microbial diversity, mainly bacterial diversity? Well, we decided to take a measure of species diversity within our sample. Some of you might be familiar with a thing called alpha diversity measure. And what the alpha diversity measure does is it looks at your samples. So in our corn soybean sample, what is different within the sample? In our, canola, in our corn soybean, what is different within the sample? And soybean, what is different within the sample? So the higher the value you get, it means you have a higher number of unique species, which is good because the higher number of unique species you have, it means you have the tendency of having a higher number of beneficial species that could be good for the plants. And so inversely, the lower value you have, it means you have fewer unique species detected in your samples. And as we can see on this box chart with the whiskers and all, the corn soybean sample had the least value of alpha diversity, which means it had the least diversity measure. Again, we used R, to determine if this was significant, and the differences were significant. Then we can conclude that corn alters soil bacterial diversity, and not only does it alter it, it carries over in a winter season. This is very essential for those of us in this part of the country, in this part of the world, because a farmer would want to know that if I treat my plants with a certain kind of microbes, would it remain the next year, or do I need to treat it again? And so this was something we were able to determine based on the alpha diversity measure. Next, we measured another kind of diversity. It's a beta diversity measure. And what the beta diversity measure tells you is how different are your samples from each other. All of these, like I said previously, were done using KIME, which is a powerful bioinformatics platform that some of you might not be familiar with just because you're in the IT stream of things. But those of you who are microbiologists or even doing just a little bit of biological data, either human biology or plants or any kind of animal biology, highly recommended tool. It's free, so it's an open source, lots of tutorials, you can learn it yourself. I think it's going to come in really handy for you guys. You can get tons of data and power through things. So with the beta diversity measure, we were able to determine that our corn soybean sample, again, was much different from the canola soybean and the soybean sample, where the bacterial population in the corn plot was a lot lower. And so with this, you guys can be wondering, okay, you know, why should we care about all of these things that Ayo is spilling out today? Well, it's very important for a farmer who once grew corn on his crop. So let's assume I'm a farmer and this year I want to grow corn. All I need to know is it's okay for me to grow the corn, but next year, before I grow my other crop, either soybean or canola or wheat or whatever it is, I need to do something to my soil. I need to add an additive because that corn has robbed my soil of some essential microorganisms that needs to be replenished. Next, we looked at the community abundance. So what's the abundance in this plot? And to do that, again, KIME, sorry I keep throwing KIME at you, but these are the tools we use. This is the tool I used for this. KIME was used to assign taxonomical classification to all of these samples, where just for the purpose of this talk, I thought leaving things at the phylum level will give you guys a sense of what is happening. So we took samples at different stages, but I'll focus on snapshots of things where before anything went into the crop, so into the ground rather, preceding, we looked at what the community composition looked like. And as you can see from the graph over here, not that much of a difference. They all seem to have equal value of things, equal value of acidobacteria. All these are different types of bacteria that I wouldn't bore you with the details for this talk. Then at the middle of the growing season, which is your V6 stage, that's the boom, middle time of growth, we see that things are starting to change, where the corn is having more of certain species, but the soybean and canola samples are still having similar values of certain species. And then at the end of the growing season, we wanted to see what things look like. Again, the corn had done, it had changed things up, whereas soybean and canola had left things the way they were. And so the bacterial diversity in the corn plots, we can definitely say are different than canola and soybean plots. So all of this information was gathered from all the amount of data I had told you about previously, and we were able to draw certain conclusions. Having said all of these, what can we do with all of this information? It's cool. As scientists, we're excited. We like to see what things are. We like to see what things work. But how would the average person walking on the streets, like how does this impact their life? Why should they care? Well, this brings us to our second research question, where we want to know if we can go to this land or to this farmlands, isolate certain microbes, and if these microbes can be used to improve crop performance. So I would show you a little bit of the data and how we did all of that, where we had our negative control. So this is a crop that wasn't treated with anything. This is treated with isolated bacteria, AB1, because I isolated it so I get to name it. One of the perks of science, when you isolate something that seems to be a bit novel, if you like, you can name it after yourself. Nobody would, blame you for doing that. And isolated bacteria, AB2. See how these bacteria have been able to not only increase your root density compared to your negative control, they've also increased your shoots compared to your negative control, and we had a positive control. And all of these are essential because as a farmer, I want to know how quick can I get results. So give me tools to increase my yield, but decrease the time frame. And from these, we're coming to the conclusion that all of these bacteria can be used in place of fertilizer. Your crops will reach maturation quicker, you get a quicker yield, you get a quicker turnaround, you can grow other crops and make some more money. So we didn't stop at this. We took it a step further, where we wanted to know what all of these bacteria were doing to the plants at different levels. So at biological level, cellular level, and other levels. And for this, we came up with this very nice schematic flow or workflow, where we've isolated our bacteria, we treat the plants with the bacteria rather, and then we analyze our samples at different time frame. But for this talk, I'm only going to show you results that we obtained at 24 hours, so 24 hours post introduction of the bacteria, and 7 days post introduction of the bacteria. You harvest your samples. Again, you prepare your library, you do next generation sequencing, and then we analyze the data. With these, we're only going to focus on the gene ontology that were enriched. So genes that showed highly regulated patterns, genes that prior to the introduction of the bacteria, they weren't so expressed. Then the moment we introduced the bacteria, expression levels went up. So like I said, we divided them into different groups. You got your biological process, your molecular function, your cellular components. But because I know most of you are familiar with photosynthesis, we'll focus on what the photosynthetic results look like, where I'll walk you through it. Could seem maybe busy for those of you who aren't familiar with this, where we looked at our root sample 24 hours after we inoculated the plant or after we introduced the bacteria. And this square means the lighter the square, the lower the expression, the darker the square, the higher the expression. So we see that for photosynthesis in the roots, which is not surprising, at 24 hours post-introduction, you don't get that much activity. But at 7 days, the activity level has increased. Same thing in the shoots, where 24 hours you don't get much, but 7 days you get much. And that's because by day 7, the bacteria has already had enough time to interact with your plants. And we also have other photosynthetic apparatus and photosynthetic processes that we measured, and things were consistent majorly all through, where you see an increase after the bacteria had had the time to colonize the plants in all of these processes. And this is good because plants need photosynthesis to grow, right? So we're showing that our sample increases the photosynthetic process, which means plants will be able to grow quicker, again, giving a farmer or those in the agricultural industry what they really want. So we can conclude that the isolated bacteria may increase photosynthetic and nutrient uptake capacity of the plants. Now, this is all important because from what I'm telling you now is, in fact, if I were to present you with things, with a sequence of things, instead of showing you how logical things are, we did this bioinformatics process. So we did this data science process before we did this. So we went into our samples, looked at what made sense. Again, we could do all of this quickly because we had all the algorithms and we figured things out by using data science. And then we went back to the lab and tested it. And that's pretty much what bioinformatics or data science does for you. It makes you look at things from a big perspective. Instead of you going into the lab and trying to do all of these experiments by yourself, these things take time. You're going to just be wasting time. Go from your big data sets, delve into it, use all these algorithms that just take a couple of days or sometimes even a couple of weeks. Focus on what you want to focus on, excuse me, and then go into the lab and start testing things. And that's really the idea I want to leave with you. I want to leave with you guys today is when we're using data science tools in biology or in, excuse me, or in the sciences, it just helps to make our life easier. It doesn't mean we're not going to go back and do those experiments, although some people only focus on the data science things and they publish and they do all of that. But for we who also want to apply our results to real life, we go back to the lab, do it, and then we take all of these things to the field or take it to the market, just to show you guys that bioinformatics/data science helps us to crunch the time by a reasonable amount of time. So from all of these, we can pretty much conclude that using data science, crop rotation impacts the soil microbiome, so the bacteria population, like I told you guys previously. Not only that, using data science, crop rotation impacts the diversity, where we can see that the corn sample, screwed up with the diversity, whereas the soybean and the canola samples were quite okay. Then the corn rotation may benefit from a boost. Like I said previously, a farmer, for example, should know that after planting corn, it would be nice for you to replenish the amount of beneficial bacteria you have in your soil before you want to plant another crop, just because the corn has used up almost all of that. And then lastly, all of these are applicable. All of these are really essential because they provide new tools for the agricultural economy. And it would also be nice for you guys to know that most of all these projects that I'm talking to you guys about is in conjunction with industrial partners. So companies are paying for us to do this research for them just because they know that once we can capitalize on this product and take it to the market, they would make profits from it. That being said, Gbemisola would like me to show you guys just a little bit of how I do some of these things. And so that's what we're going to do now. Sadly, some of these data are confidential, so I can't use my work computer. And I've installed a virtual machine on this computer. What that's going to allow me to do is to log into a Linux operating system and show you guys a little bit of what we do using KIME. And I would like to say up front that KIME uses Python. So if you're a sucker for Python, this is your place to shine. This is your place to shine. So I wouldn't run all of this code because if you're familiar with things, you would observe that my computer, my small laptop, has two operating systems. It would crash if I were to run all of these simultaneously. So do the Zoom and run all of this script simultaneously. So what I did was I ran most of them yesterday and 2 days ago, and I will just show you the kinds of results you can expect to get. If you're someone who likes using Jupyter for things, it's not bad. You can use Jupyter for KIME too. All you need to do is install this extension. So I found this online. I don't use Jupyter. I do all my coding on Linux using this terminal right here. As you can see, I ran a few not too long ago. But if you're really in for Jupyter, it's also good. You have all these tools incorporated in notebooks and whatnot. So what you do is
Sorry, Ayo,
One day.
is your
Yeah,
slide
yes.
that we're still saying no?
We're
I don't
still
know if
seeing
you're
my
showing.
slide.
Yeah, I don't know if you're showing something else. You
Okay,
can
let me. Yeah,
stop
yeah.
sharing
I
and
know what I'm doing.
okay.
There we go. Can you see my
Yes.
was? Yeah,
Yeah.
you can see something different. Okay.
Yeah.
Sorry about that. So yes, so this is the extension I was referring to that you can install in your notebook if that's something you want to do. So you install this, you can see the KIME code right here, install it, you'll be fine. And once you're done doing all of that, you also need to install the necessary modules. All of these will give you the necessary modules you need to run KIME. Again, I would like to say that most of these, in fact, all of these can be found on the website where you can download KIME. Once I'm done showing you guys all of this, I'll show you where to get all these resources too. So those are for people, that's for people who want to use Jupyter. If you just want to install KIME right away on your terminal, so if you use Linux, all you got to do is do that and you have KIME, you're ready to.
Hello.
Oh yeah, hi Olumide.
Sorry, you said I just want to know the notebook that you use for this, your queries. You mean Jupyter? This doesn't look like Jupyter, please.
Yeah, this doesn't look like Jupyter, and that's what I said earlier. I do most of my coding on Linux, but I'm aware that, so I use Linux. Yes, but I'm aware that a lot of people use notebooks. People want to use Jupyter, you want to use all of that. And so I thought it would be nice to show you that you can use notebooks for that. And that's why I included this before I get into the nitty-gritty of things. So if you want to use,
I actually joined late. So were you able to use also some library application to your studies, or you were just writing the script?
So yeah, so I write script.
Okay, sorry, Ayo. Olumide, if you can just type your questions in the chat box so we can take everything together, if that's okay. Yes, please. So type it in the chat box so she can quickly go through it and then we'll take all the questions together in case someone has a question similar to yours as well.
All right.
Thank you so much.
Okay, yes. So good questions, for sure, good questions. I write scripts just because I like the challenge it poses. I like to remember things, but that's just me. If you would rather, do things just like what Olumide suggested, go for it. All of these resources are online. You just need to find what's going to work for you. So I write my scripts and that's just me. That's about that. So we're going to be working, or I'll show you results from data sets on KIME's website, where what this study did was it looked at two individuals, took microbes, so microorganism swab from different parts of their body, 5 different parts. And what they wanted to do is they wanted to see if the microorganisms cluster together, so if they relate together regardless of the sample. So let's say they take a microorganism sample from me and they took from Wemisola, two different locations, but they want to know if these microorganism sample overlap regardless of the individual. So that's what we're going to be powering through today. I'm not going to spend too much time on things, but just to give you a sense of what it ends up looking like. So you download, you make your directory, which we all know that's very essential. Make your working directory, make sure you're in your working directory, and then you get your metadata file. If I was working with my own sample, I would make my metadata file. So a metadata file is a file that tells you more about your data. And this is what their metadata file looked like, where, like I said, they obtained different microbial swab from different parts of the body, 5 parts: the guts, the palms, left and right, tongue, gut, and all of that. They took the sample in a span of a year, and they just put things in their metadata file, categorized it as either numerical variable or categorical variable. There we go. So you want to make sure you have this file downloaded on either your Linux or your notebook. You're going to need your metadata file. Let me go back to the notebook. So you get your metadata file, either you download it online or you tell your terminal where to find it. You also get the sequences. So like I said, the sequence, we work with sequences. all our data as sequences. And this is what sequences look like. You just get a blob of things where if you're a scientist, some of it will look familiar. All of these are your nucleic acid, so things that end up making your DNA. And the other things are just more information about how your data was sequenced and all of that. Yeah, it would also be nice to see that this file is gigantic. Look at where my cursor is. You get a line of 1.2 million lines just of information. All of these things are crazy, big data, but we can try to make sense of it and get what we need from all of that. I'll go back to the tutorial, the notes I made. So once you have your data, your metadata and your data and all of that, you import all of that into a KIME artifact. KIME, like I said previously, and I'm going to keep saying it so that you guys kind of know what I'm talking about and you're familiar with it. I also wasn't familiar with KIME until I started doing all of this kind of work. But it's very important for not only plant biologists, if you're working with human biology, like I said, or plant biology, or even animal biology, it helps with getting things in check and also assigning species name to your bacteria or to your fungi. It also does work with fungi because a lot of pipelines have been built for it and a lot of data have also been loaded onto it where almost all the bacteria in this world. So there was I don't want to delve too much into it, but there was a project where a group of scientists wanted to sequence all the bacteria in this world, which they've done a substantial amount of work on that. So all the information they obtained has been loaded onto KIME. And that's why I think it's a powerful tool for things like that. So when you get all your data onto wherever it is you want to do all this, your programming / data science analysis stuff, you need to do some quality control. So we call it quality control, where you clean up your data. If some things don't look right, you clean it up. And what I use is called a deblur, where if we see, so we look at this data. So this is the data we're working with, where the quality score is high. And then when you get to around 110/120, it drops. So as a biologist or as a bioinformatician, I don't want to include all of this in my analysis just because it's useless. The quality score already dropped. It's not going to give me a good idea of what is really happening. So these are things where you call background information. I want to get rid of all of this background. That's where quality control comes in. In fact, you almost have to always perform quality control on all your data. So we do the quality control step using the deblur, like I mentioned, where you do all of this. So we visualize, that was the visualization code used to visualize. Then you come over here and do your deblur. So I decided to trim it at 120 length just because, like I showed you previously, everything before 120 just didn't make sense. I could do it at 110, but just to be safe, you're like, oh, maybe I'm going to miss a few things. It's always nice to just go a little bit further. So we're going to trim it at 120, which was what we did by simply using this code right here, the deblur code. Once you spend a few times on these software, you get used to all of these things. Or on even KIME's website, it tells you what each of them do, and you're able to move on with that. So once we do the deblur to clean up our data, we can then visualize it and see what it looks like after cleaning it up. And so I told it to visualize it producing a table just because I didn't want to go through things anymore. So after that, you're left with 770 sequence counts, which is still okay. And you can see that all the length of our samples are 120. So all the length, all the sequence length, you go from the top, you scroll from the top to the bottom, are all 120 because it got rid of anything after 120, since we found out that that wasn't really going to be useful for us. Again, on KIME's website, they tell you all the tools you need. So there's this thing called Empire, which I loaded already, and it helps you to visualize your data on a very nice 3D perspective. That's one of the reasons why I keep going back to KIME. It has everything already pre-installed. All the plugins you need are already in there. So we want to see how different our samples are from each other. We do our core matrices, where you use your diversity pipelines. You use a phylogenetic rooted tree. Biologists will be familiar with all of these. Choosing your sample depth kind of comes with experience, and it's something you have to play around with. But I chose a 1,109 sampling depth because that is what worked for this data set, and that is what gave me the representative sample that I thought was good enough. And you want your metadata to be the metadata that we've been working with previously, which is this over here that I showed you already. Then you load all of this, you run it, and then you load your Empire plugin and use it to visualize your data, and you get something like this. I'm actually going to spend some more time on this. So this is the Empire or Emperor thing, where it also has a place for notebooks. So if you're into Jupyter and all of that, it takes you to your notebook view, but we're not going to do much on that. But with Empire, you get something like this, where first of all, it moves around, which is cool, but then it clusters your sample, where you can see the red, which is your gut samples, clustering together, the tongue, which are your green samples, clustering together. And I'm going to say it out there, every biological data has outliers. So outliers are samples that don't perform according to your norm. So you have few outliers, but we're going to focus on the majority of things. And as expected, your palm sample, regardless of where they were obtained, either your left hand or your right hand, clustered together. So now I'm going to change your sample. So like I said, I'm going to change your subject. This was taken from 2 individual. Regardless of your subject, things cluster together. So you see how your subject 1, so individual 1 and individual 2, both their, I think this was the gut, clustered together at the same place. Both their tongue samples clustered together at the same place. Both their palm samples clustered together at the same place. So it didn't really matter who these samples were taken from, rather, all the samples clustered together. And that was good for this study because I'm pretty sure it was the first to show that bacteria don't discriminate based on individuals, they discriminate based on body size. That's very important for microbiologists. So I'll go back to my presentation. Let me share this screen. I'll go back to my presentation and show you guys what I want to leave, like the message I want to leave you with. So let's go back to that. So all of this is important because there's an explosive growth in the amount of biological data that are out there, not only in plant science, even in human biology. In fact, I've heard of, or I've even seen job postings, and I've heard of people who work with data that have been collected since the 1970s, HIV data, polio data, that they want to make sense of it. They need bioinformaticians, those that have knowledge of all of these things to power through, because these data are a lot. You can't do it manually. So all of these tools will help you with that. Then there's also the issue of data mining, where the process by which we have all of these hypotheses need to be tested and are generated regardless of the structure of the gene or protein. So like I said, we scientists tend to deal with hypotheses. So you have your hypothesis out there. You can use data science mechanisms to test this hypothesis. Then we want to start looking at things from a more global perspective, instead of looking at things from an individual point of view. So how does this one organism affect this one organism? We want to take things grand scheme. And when you take things on a grand scheme level, it also means you are working with more data. And so data science is a tool that would help you to look at things from an whole organism approach instead of from an individual approach. So my parting words and the resources I will leave you guys with is R&R Studio. This link does a good job telling you how to download R, how to make use of it for your data sets. R also has a lot of data sets loaded onto it that you can download and use for your stuff. These network analyses I made using R. I thought it looked cool. It has nothing to do with data science. I'm going to say that out there. It's actually HIV data. Very cool. If you want your presentations to have very, very good images, R is the place to go. KIME, that I just spent, majority of my presentation talking about, can be downloaded. Everything about it is right here, kime.org. Then Coursera is a platform that offers tons of courses. I have taken a few courses via Coursera, and for those of you who are interested in a little bit of what I have said, I will recommend this genomic data science specialization offered by Johns Hopkins University. The beautiful thing about Coursera is enrollment is free. You only have to pay if you want certification. I never paid for any of my courses. Just take the course. It teaches you so many things. This course in particular goes through next generation sequencing experiments. So all the things I told you we did, it breaks it down for you, does everything, DNA, RNA, epigenetics patterns. So let's even assume you're not a biologist, but you somehow want to get your hands on all this biological data. Start from here. It's going to teach you how to use bioconductor, biopython, so Python that are pretty much essentials for real biologists. The pipelines and all of that is going to introduce you to that. Then it's going to introduce you to Python programming language itself, then programming in R. I do some of my programming in R. It's very essential. Galaxy for RNA sequencing data, it's huge. It's very nice. If you can make the time to follow courses like this, it would really, really help enhance the tools you have in your toolbox. And lastly, I would just like to tell you that you have to understand your strength and build from that. I, for example, am a plant biologist. That is my strength. I'm building on that. So I'm not just going to try to apply data science to something I don't know without doing my due diligence. So like I said, even if you're not a biologist, go do your due diligence, read up on things, and then build from that. Then you have to be comfortable with teaching yourself. Hardly do you have somebody sitting with you or a teacher sitting with you and teaching you things that are outside the curriculum of a program. So you have to be comfortable with teaching yourself. You have to invest some time in yourself, use data sets online, those that are in your domain, those that are not in your domain, because all of these things take practice. The more you do it, the more comfortable you are with yourself, the more comfortable you are with a lot of things. And lastly, I do this a lot. Google, spend so much time on Google. You're going to get tons of information that will end up being super useful for you. And yeah, I think I've spoken enough. And any questions you have,
Oh, wow. Ayo, that was amazing. Please, can we just say a big thank you to Ayo in the chat box, please? That was amazing. Like, you know, even though I'm not from plant biology, but I think I really personally enjoyed, you know, the journey that you've taken us through, helping us to understand what you were trying to do in plant biology and how you were able to apply data science to solve it. So I can see people responding already. So again, please, if you have any questions, please put it in the chat box. But before we go to our individual questions, we have some pre-asked questions.
Okay, excuse me. Do you want me to stop sharing my screen, or it doesn't matter?
Yes, I think, well, yes, actually, I think I can actually share without you stop sharing. So I think it's fine.
Okay.
Yeah. So can you see my screen?
Yes.
Okay, so we have a few. Oh yeah, I can see people saying, "Oh, thank you, Ayo. That was wow. That was very insightful." So thank you for your comment.
You guys are welcome. Thank you for coming today. Thank you for this platform. I really enjoy talking about my work, and just to let you guys know that you don't have to be an expert in everything. It's okay. Yeah, it's okay.
Yeah. Okay, so I'll just go through the questions one after the other. And so the first question says, how can data science be applied in plant biology? Can this application be related to bioinformatics?
Oh, yeah, well, I think we touched on that during the presentation.
Yeah.
Data science can be applied to plant biology pretty much by all what I did. So going into the field, getting your plant samples, isolating bacteria, doing your next generation sequencing, and doing all of that. Then bioinformatics, like I said, is usually used interchangeably with data science in our field. It's used interchangeably. So it's pretty much what we just spoke about.
Okay, then is there any relationship between data science and biochemistry? That's the second question.
Yeah, oh yes, tons. So let's take a pharmaceutical company, for instance, who just designed a drug and they want to know the activity of this drug on how it affects human beings. I'm pretty sure that's a little bit of biochem because it's a chemical and you want to see how it affects biological function. A study like that would rely on more than one or two individuals, right? You need a lot of individuals just so you get a representative of your population. So with that, you can use some data science tools to power through whatever data you get from there And just like the example I went through using KIME, so the data that was loaded, you can see how, let's say, a drug affects cancer cells, different type of cancer cells. So how does it affect a sample that was taken from a cancer that grows on the skin versus a cancer that grows, let's say, on the tongue? And you can just try to make sense of things. So it doesn't really matter what your discipline is, as long as the main issue is having a lot of data that you need to power through. I believe data science can be applied to that.
That's so powerful, Ayo. Thank you so much for that. Then the third one, this must be someone related to plant biology as well. How can
Right.
microbial community structure studies how can it be
Apply in
applied
the
in the
field. Okay.
field?
Excuse me. Well, I think I touched a little bit on that too in my presentation, where we showed that the community structure of corn was different from that of canola and was different from that of soybean. So if you want to apply this to the field, which means you want to apply this to a real life example, to an agricultural field, like I said, all you got to do is talk to a farmer or talk to corn growers that, guys, it's okay that you're planting your corn, but just know that once you're done planting and you need to harvest your corn, based on our microbial community structure studies, you need to do something to your soil. So you either need to replenish it with more bacteria before you grow your other crop, because we know that corn has screwed up with things, or you need to buy our products, which is going to have all of these good and beneficial bacteria, and use that to enhance your field. So that's one of the applications that relates to what I just said, that will work from that. Another one that might be of interest to this person, just because it seems like they know their plant science stuff, is from microbial community structure studies. We do this too, I do this too. You can isolate your good bacteria. So those bacteria that I named after myself, what I'm doing right now is I'm sequencing their genome. So I want to know what the genome of each bacteria looks like. Why is that important? Well, if I see that this bacteria produces some antibiotics gene, or is able to encode antibiotics gene, or is able to somehow encode a plant growth promotion gene, that alone, is sufficient to be able to apply it to the field, where such a bacteria can be used to make products that I can coat seeds with. So you can coat your corn seed, your canola seed, your soybean just with that bacteria and sell it to farmers. So I think that's that's a more applied approach to of how all of these things can be used.
Okay, thank you so much, Ayo. Then I'll take, question 4, so that we can quickly wrap up. It's already 5.
Okay.
How long have you been working on big data sets, and what advice would you give anyone new to all this without a science background?
Well, full disclosure, I've only been doing all of this since January, so 6 months. And the advice I would give anyone new to all of this is what has worked for me. Research and investigate things. So Google, like I said, Google, make the time to teach yourself. All of these resources are out there. Majority of them are free. So these are things you have at your fingertips, free resources you have at your fingertips. Make the time to learn, cultivate the habit of reading stuff, trying stuff yourself. You'll be stuck, that's for sure. It hasn't been that easy. It's been a huge, learning curve, but power through it. And if you're new to all of this without a science background, so let's say your background is social science or the arts, I'm pretty sure websites like the UN, WHO would have data sets you might be interested in. So you could have data sets of the number of homeless people in a city where you can work with. So for you who aren't in the science field, that could be something you're interested in. You want to know what's the homeless rate in a certain region, and how can you apply such information to, let's say, policy making. So you can work from that, go to all of these websites, get the data and start working through it. Watch YouTube videos on how to start. So if you really have no clue of where to start, watch YouTube tutorials, start from there. And I think data science also has its place even in non-scientific fields. So art and, social sciences, even though social science is a little bit of science, but I think you get what I'm trying to say.
Thank you so much, Ayo. I'll combine the last two questions because they're similar. Someone
Okay.
says, what's a step-by-step course to take as a beginner in data science? And then question 6 says, what specific courses
Oof.
should one be looking to take to become an efficient data scientist in no time?
In no time. Well,
In no
everything takes time. So
time.
that I would say. But the course I presented earlier, the one in Coursera, is a good place to start for those who have a little bit of scientific background or who want to incorporate some science into what they are doing. So when I say science, I mean biology, biochem, microbial, and all of that. But if you're someone who just wants to use your data science stuff for IT and want to continue through that route, Coursera has more. So it's a platform where all you got to do is, yeah, so somebody says start from MS Excel. That's good. Of
That's
course,
true,
very good.
actually.
Tons of tutorial on MS, and I've said MS Excel, and I've said this, I say this to so many people. Excel is so underrated. It can do so much more. So if you're someone that you're like, you know what, everybody kind of knows Excel. That's what I want to make my own niche. Start from there and build on it. Become an advanced user of Excel. There are tons of resources on Excel too. Tons of tutorials on YouTube. Tons of courses too. Free courses on Excel. Start from there. If you then want to grow on it, like I said before, just Google things. You could Google what specific course should I be taking to be an efficient data scientist in no time. It will tell you. There are people who have reasoned like you, and there are people who have answered questions like that.
Yeah, thank you so much, Ayo. So definitely Excel is a place to start. And people always talk about learning, like if you want to choose a programming language, Python is one easy one to start with as well. Even though our last webinar, we had a huge debate around Python and R and which one is
Yeah.
better. But I
I
mean,
love R. I love
yeah.
R. I know it's not for
Yeah.
everyone.
So you can try both and see which one works for you. I'm not familiar
Exactly.
with R. I only know Python, and so and I've enjoyed using it. Even though I'm not a data scientist, I apply Python as a DevOps engineer. So that's because I haven't actually even tried R. So sometimes I would also say, as far as IT is concerned, it's very important to get your hands dirty. I always say that a lot. That's how you know what you're comfortable with, what you really like, what tool gels with you, what tool works for you. That's the best way to find out. So thank you so much once again, Ayo. We've answered the pre-asked questions. Does anyone in the audience have any questions? You can actually raise your hand if you want to speak. And then, I don't know, I think you can unmute yourself actually if you want to speak or ask any questions before we wrap up. It's, okay. Muiwa says, to add, Coursera also has some courses in Excel and Power BI. Yes, we talked
Correct.
about
Yeah.
Power BI too in the last webinar. So surely check Coursera for the kind of courses that you want to do and what you would like to learn. Thank you so much for that, Muiwa.
Hi,
Okay.
can I quickly say something?
Sure, please talk. Sorry, I didn't know your hand was up.
No problem.
Yeah.
Just to I'm too tired to type. With regards to I know I joined really late, so sorry about that. With regards to where to start from, there's some
Yeah.
like what Ayo and Gwemi said. There's some courses in Coursera, and also for if people are interested in data science, to be exact, there's a website called Udonics. I'll probably check the spelling and send it right now. You can probably start from there. It has introductory courses to data science itself, the basics, and some textbooks that people can read on.
Wow, that's amazing.
Yeah.
Amazing, Muiwa. Please type it for us. I'm sure a lot of people, even
No
people that
problem.
didn't join today, will be so interested.
Yeah.
Like,
Wait, do you know the Muiwa you're talking to?
I know now. It's Muiwa
Just to be
Tubi.
sure.
I know your
Oh,
voice.
Tubi. Oh my goodness.
Yeah.
You know Tubi.
Yeah, I know Tubi. We went to high school together.
High school? I
Yeah,
went to high school with Tubi. Okay.
Oh, good.
Was it? Is it the same Muiwa you're referring to?
Yeah. I'm sure he knows me.
Muiwa, do you know Ayo? Talk now.
I do.
But
Very.
okay.
Went
Anyway,
to school
we'll
together.
discuss offline. Which school did you go to? My school? How did you go to? How many schools did you go to? Okay.
Quite a number.
Okay.
All right. Yeah.
Okay.
Give me a second. Yeah.
Okay, yeah, just type response to that. Okay, yeah. So again, this call is very informal, so don't mind us that we're talking about school. So does anyone else have any questions before we wrap up? Anything else?
Or even
Please feel free.
suggestions.
Yeah. Any information that you feel will be useful for everyone. I mean, everyone here already has some form of interest in either IT or plant biology. So anything, if you're already good in data science, we would like to hear from you.
Sorry. Okay, am I still okay? If anybody is interested, they can actually look at GIS.
Where?
Oh,
Sorry?
GIS stuff.
Yeah.
Can you send it to the chat, please?
Maybe Gbemi Sola should have you on next, because that's very applicable too.
G what? I didn't get that.
GIS, Geographic
GIS.
Information Systems.
Oh, Muiwa is the pro in anything geo. So okay, yeah.
Amen to that.
Yeah, I'll link up with you on that one.
All right, no problem. All right, so I've sent it to the website. So yeah.
Okay. Thank you, Muiwa.
You are welcome.
Anyone else who has anything to say? Should I start calling names? Thank you everyone for joining us. It would be nice to have heard everyone, but everyone seems to be muted. I'm about to wrap up. Adeoye Shotayo, thank you for joining us. Anything to say? Ade Shola or Adesola Julius, Audi, Ayodeji Salami, Damilola, Etiosa Joy, Juwolo, Nwali, Senna, Slyman, Tolu Alashe. Okay. Anyone else? Oh, I think Olumide is gone. Olumide had a question there, but you already answered this question, Ayo, right?
Yes.
He was the one asking about why scripting and
right.
Okay, cool. So I guess we've come to the end of the webinar today, and once again I'll just like to say a very big thank you to Ayo for having us today, and for coming to this platform today. And if you would like to connect with, surely, if you'd like to connect with the Outstanding Professionals Network, please get in touch with me on any of these means. And if anyone here is a specialist in any area, you know, and you would like to possibly speak, please also get in touch with me. I mean, it's always good to share knowledge. I mean, IT is the future, tech is the future. Without tech, we wouldn't have survived COVID-19 as a whole world. So tech is the future, you know. So any knowledge you have and you feel like you're a specialist and you like to share knowledge, to be very good to bring you on this platform. So connect with us on LinkedIn, connect with us on Instagram, Twitter, YouTube, and I would also send the slides out for you to get the links to those courses that Ayo has recommended. Thank you so much, everyone, and yeah, goodbye. Thank you. Ayo, thank you.
Thank you. It's been a pleasure.
So much. Yeah. Thank you. You can also unmute yourself and say thank you. Oh, Wally says I was late, but for the brief moment, I say a big thank you. Oh, thank you, Wally. Many thanks to you guys. Okay, cool. Is that a question?
Thank you, Ayo and Bemi.
Finally.
Thank you.
Yeah, I really appreciate it. Yeah.
Thanks, Julius. Thank you.
Thanks for joining us. Thank you.
Thank you, guys.
Cool. Thank you.
Thank you.
Thanks for joining.
Thanks.
Thank you.
Thank you. Thank you for the presentation, and well done on the initiative. I watched the previous one as well, and I really enjoyed it. So keep it going. And
Well, thank you so much for that feedback. It's really good to know that we had someone who went to the last and is here again. Thank you so much, and yeah, thank you.
Bye.
All right. Have a good evening.
Yeah, have a good evening. Enjoy the rest of your day.
Thank you. Cool. Thanks, everyone.